Welcome to the deep dive. So, I want you to imagine a flower that basically looks like a botanical supervillain.
Oh, I like that description.
Right. I mean, we are talking about these massive, pale green petals that stretch over 9 centimeters across. And protruding straight out from the center is this striking, pitch black, almost velvet textured tongue.
It is incredibly dramatic. That's the Coelogyne Pandurata, or, as it's commonly known, the Black Orchid.
Yeah, the Black Orchid. But here is the crazy part. This plant is so stubborn, like so fiercely uncooperative, that it is actively allowing itself to go extinct.
Yeah, the stakes with this specific plant are just incredibly high. It's native to eastern Kalimantan in Indonesia, and it is packed with potential medicinal properties.
Specifically a ton of antioxidants, right?
Exactly, really high concentrations of antioxidants. But the problem is it acts like an absolute diva.
The total diva.
I mean, in the wild, it rarely blooms. You're talking maybe 2 or 3 times a year if you are lucky. And when it finally does produce those incredible black-tongued flowers, they wilt in about 6 days.
Which is just heartbreaking. So, for you listening, we are looking at a biological ticking clock here. And our mission today is to explore a stack of really fascinating research centered entirely around saving this one highly exotic plant.
And what's interesting is that the sources we're pulling from today tackle this crisis from 2 completely different scientific angles.
Right. On one hand, we have this deep dive into phylogenetic DNA mapping from peninsular Malaysia, basically figuring out how to save its lineage through hybridization.
And on the other hand, we have some cutting-edge biochemical research aimed at, hacking the purebred orchid's diet to essentially force it to bloom and produce medicine.
Let's start with the lineage problem, because if we don't figure out how to increase the population, the biochemical hacking doesn't really matter.
Very true. To save the black orchid from extinction, conservationists desperately need to crossbreed it and create more seeds. But orchids have these incredibly specialized, highly restrictive reproductive systems.
Okay, let's talk about those restrictions. Because honestly, my initial thought was, if you have a super rare orchid in a lab, why not just pollinate it with its own pollen?
Which is called autogamy.
Yeah, autogamy. Or, at the very least, take pollen from one flower and move it to a different flower on the exact same plant.
Right. Geitonogamy.
Exactly. But the data from the sources shows that both of these methods fail almost completely. Like, why?
Because the plant actively fights it. It employs this evolutionary defense mechanism known as self-incompatibility.
So it's rejecting itself.
Pretty much. From a survival standpoint, plants want genetic diversity to avoid inbreeding depression, because inbreeding leads to weak offspring. So over time, they developed a biochemical recognition system.
How does that actually work, though, like on a cellular level?
Well, when pollen from a black orchid lands on the stigma, which is the female receptive surface of its own flower, the plant's cellular receptors actually analyze the proteins on that pollen.
And if the proteins match too closely, what happens?
The plant sounds an alarm. It basically registers its own genetic material as a threat.
Wow. That is wild.
Yeah, and then it deploys enzymes that physically halt the growth of the pollen tube.
So it shuts down the whole process.
Exactly. Normally, a pollen tube has to grow all the way down into the ovary to deliver the genetic material. By blocking that tube, fertilization becomes physically impossible. The unfertilized fruit just shrivels up and drops off.
Which, is a brilliant mechanism if you're living in a massive, thriving rainforest population.
Oh, for sure. It forces the plant to wait for pollen from a distant neighbor, keeping the gene pool healthy.
But when you are the last orchid standing in a shrinking habitat, that same defense mechanism is what drives you into extinction. It is literally suffocating its own reproductive cycle.
It's a tragic irony. And because of that self-incompatibility, scientists had to look outward. They needed to cross the black orchid with an entirely different species.
But one that was genetically close enough that the pollen tube wouldn't be rejected, right?
Exactly. So the researchers essentially had to play detective. They gathered 59 different Coelogyne orchid samples from all over peninsular Malaysia.
59? That's a lot of orchids to sort through.
It is. And reading through their methodology, it's really cool because they didn't just guess based on which flowers looked similar. They used traditional morphological characterization.
Meaning they meticulously scored a bunch of physical traits. 30 different ones, actually.
Right. They analyzed the architecture of the plant. They measured the shape of the pseudobulb.
Like pseudobulbs, those are the thickened stems orchids use to store water and nutrients, right? To survive dry spells.
Yep. Exactly. They documented the shape of those, plus leaf symmetry, the type of root systems, and the structure of the petals.
But, I mean, relying on physical traits alone seems a little risky. Like, two plants might evolve similar water storage bulbs just because they live in similar climates, not because they are actually closely related.
You're spot on. Convergent evolution is a major hurdle in taxonomy, and that is why they paired the physical trait scoring with modern, highly precise DNA sequencing.
Right, they brought in the heavy machinery, and they focus on a very specific genetic marker called the ITS region.
The internal transcribed spacer of the ribosomal DNA.
Okay, let's unpack this because the mechanism here is so fascinating to me. Ribosomes are basically the protein factories of a cell.
Yep, they build everything.
And the genes that code for ribosomes are so absolutely critical to survival that they rarely mutate. Like if a plant mutates its ribosomal genes, the protein factories break and the plant just dies.
Exactly. But what's fascinating here is that the ITS regions are the non-coding spacers between those critical genes.
So they don't actually build anything.
Right. Think of them like the blank pages between the chapters of a book. When the cell transcribes the DNA, these spacer regions just get clipped out and discarded.
Because they don't actually do anything vital, which means mutations can pile up in those spacers without harming the plant at all.
You got it. And that creates a high-resolution evolutionary clock. The core genes stay identical for millions of years, but the ITS spacers mutate rapidly.
So by sequencing these spacers, scientists can read a genetic barcode that reveals exactly how recently two distinct species shared a common ancestor.
Exactly. And when they combined the 30 physical traits with this ITSS barcode, a clear winner emerged out of the 59 candidates.
The perfect match.
The Black Orchid's perfect partner was a species called Coelogyne rumphii. The data logged an 87% morphological similarity.
Okay, hold on. Let's be real for a second. Because looking at the visual descriptions in our sources, this requires some explanation.
Uh-oh, what's the issue?
Well, the black orchid has huge, pale green petals and that giant black tongue, right? But the sources say C. rumphii has small, light yellow flowers with a brown tongue.
Right, they look very different.
Very different. Visually, this is like saying a Great Dane and the Chihuahua are an 87% match. How does that make sense?
It's a great analogy. The visual disparity is definitely striking. But evolutionary closeness isn't expressed in the flashy outward aesthetics.
Oh, so the petals are just for show.
Exactly. Petal color often evolves rapidly just to attract a specific local insect pollinator. The 87% similarity is actually buried in the foundational vegetative traits.
You mean like the structural bones of the orchid?
Exactly, the structural bones. Both species share a sympodial growth habit.
Meaning they grow sideways instead of up, right?
Right. Instead of growing vertically from a single central stem like a pine tree, they grow laterally. They send a rhizome horizontally across a tree branch or the ground and shoot up new lateral stems side by side.
Oh, that makes sense.
Plus, both species produce lanceolate, spear-shaped leaves, and they both have 4 pollinia.
Pollinia being those dense, waxy little packets of pollen that are unique to orchids, right? Not like the loose dust you see with pine pollen.
Exactly. So going back to your analogy, when you strip away the color and the size of the petals, the biological mechanics and the skeletal architecture of the Great Dane and the Chihuahua are virtually identical.
That is mind-blowing. And I guess that structural compatibility gave the scientists the confidence to actually attempt the cross.
It did. So they took the waxy pollinia from the male C. rumphii, which, by the way, is a highly energetic plant that blooms almost every month of the year.
Very different from our diva black orchid.
Very different. And they transferred that pollen to the stigma of the female black orchid.
And the result was a massive success, right? The self-incompatibility alarms did not trigger.
Nope, the plant accepted it. The pollen tube grew, the ovary swelled, and fruit maturity was reached in just 158 days.
Which is incredible.
Yeah.
And then they harvested the seeds and cultivated them in a lab, and the protocorms emerged in only 22 days.
Yeah, 22 days is remarkably fast. And just for context, a protocorm is a specialized mass of cells that forms right after an orchid seed germinates.
Because in the wild, orchid seeds don't have any nutrient reserves, right?
Exactly. They require a symbiotic fungus to infect them and provide food just to reach the protocorm stage. So getting them to emerge in 22 days in a sterile lab environment using nutrient agar is exceptionally fast.
So they basically secured the genetic legacy. By crossing these two, they created a new hybrid variant that captures the stunning black-tongued aesthetic of the mother, but injected it with the vibrant, year-round blooming energy of the father.
It is a brilliant conservation strategy, truly. But it does leave a rather glaring issue on the table.
Right, because we saved the hybrid, but we haven't actually solved the problem for the purebred original.
Exactly. The native black orchid is still sitting in eastern Kalimantan, refusing to bloom and slowly dying out.
So the question is, how do we optimize the mother species? How do we force it to produce those valuable medicinal compounds and keep it alive without permanently altering its DNA?
And this is where we pivot from genetic matchmaking to hacking the plant's metabolism.
The diet hacking phase.
Yes. The second batch of research we looked at focuses on forcing the pure black orchid to bloom out of season by aggressively manipulating its diet.
The researchers wanted to see if they could trigger a massive biological response by adjusting a single macronutrient.
Right. So they set up an experiment using foliar sprays.
Which is basically liquid fertilizer applied directly to the leaves and the pseudobulbs, right?
Yep. And they used monopotassium phosphate, or MKP for short. They tested a gradient from a 0% control group all the way up to a highly concentrated 36% solution.
And they specifically targeted phosphorus. Why phosphorus?
Because of its role in cellular energy. It all comes down to ATP.
Adenosine triphosphate, the energy currency of every living cell on Earth.
Exactly. The P in ATP is phosphorus. The molecule has 3 phosphate groups acting like a tightly coiled spring.
And when a cell needs energy to grow, it basically snaps off that third phosphate group, releasing a huge burst of power.
That's the mechanism. So by supplying the plant with exogenous phosphorus through the foliar spray, the scientists were essentially providing the raw material for the plant to build millions of these ATP springs.
They were turbocharging the orchid's energy grid.
Exactly. And the results were shockingly specific. The data isolated a definitive Goldilocks zone at exactly 14.4% phosphorus concentration.
14.4. That is so precise. What happened at that exact concentration?
The black orchid underwent a dramatic transformation. It bloomed in just 47 days compared to the sluggish 74 days in the control group.
Wow, almost twice as fast.
Yeah. It averaged over 5 flowers per stem, and physical measurements showed these flowers had the longest sepals and petals recorded in the entire study.
So the extra ATP fueled increased photosynthesis, giving the plant this massive energy surplus. But what truly fascinates me is how the plant actually used that surplus.
Right, because the energy didn't just make the flowers bigger, it fundamentally altered the plant's hormonal balance.
It triggered the gibberellins.
Right. Yes. Gibberellins are a class of plant hormones that govern development. When the energy grid surged, it ramped up gibberellin production, which sent a powerful chemical signal to the plant's meristem.
Stem cells of the plant.
Right. It forced the meristem to stop producing vegetative leaf tissue and immediately switch over to reproductive flower tissue.
So the phosphorus literally hacked the biological clock. That's amazing.
It really is. But the visual bloom was only half the goal. The researchers were also tracking the medicinal value of the plant.
Specifically the phenolics and flavonoids. Now, we often hear these terms marketed as antioxidants in human supplements, but what function do they actually serve inside the orchid itself?
Well, in a plant, phenolics and flavonoids are secondary metabolites. They are a core part of the plant's chemical defense system.
Defending against what, exactly?
They protect the cellular tissue from environmental stress, UV radiation damage, and herbivore attacks. But synthesizing these complex molecules takes an immense amount of energy.
Oh, I see. So a struggling, starving plant won't produce them because it is prioritizing basic survival.
Exactly. But because the 14.4% phosphorus spray gave the orchid an ATP surplus, it had the luxury of stockpiling its defenses.
So the researchers documented a drastic spike in phenolic and flavonoid content.
Yes, concentrated heavily inside the bulbs and flowers, which translated directly to a significantly higher overall antioxidant capacity.
Meaning, we managed to wake the plant up early, make it physically larger, and pack it with medicinal compounds, all with a single precise chemical trigger.
It's a huge win.
But naturally, when I read a result like this, my mind goes straight to the extremes.
Uh-oh. I know where this is going.
Hear me out. If applying 14.4% phosphorus is like giving the plant a perfectly calibrated double shot of espresso, it wakes up early, looks amazing, is packed with antioxidants, why not give it 5 shots of espresso?
Because that never ends well.
Right. Why didn't they just flood the system with the 36% phosphorus spray to generate an astronomical level of antioxidants?
Because plants operate on a very delicate biochemical equilibrium. When the researchers pushed the concentrations up to 28.8% and 36%, the entire system crashed.
Yeah, looking at the data, it paints a picture of a severe biological backlash.
Very severe. At 36%, flowering was delayed all the way back to the original 74 days. Fewer flowers emerged, they were physically stunted, and the antioxidant levels just plummeted across the board.
The plant suffered an overdose.
Exactly. And the mechanism behind this crash is a phenomenon known as nutrient antagonism.
Okay, let's visualize this antagonism, because the concept is really interesting. In the roots and the cell walls of the leaves, there are specialized transport proteins acting like doorways.
Right.
And if you imagine the vascular system of the plant as a highway, these doorways are basically the on-ramps. When the scientists sprayed the 36% solution, they essentially sent 10,000 phosphorus delivery trucks to the on-ramps all at once.
And those phosphorus ions physically overwhelmed the transport proteins. They just completely clogged the doorways.
And this is where the antagonism happens, right?
Yep. Because phosphorus and potassium often compete for the exact same pathways. By flooding the system with phosphorus, the scientists created a massive traffic jam that completely blocked the potassium ions from entering the cells.
Oh, wow. So even though the foliar spray was monopotassium phosphate, meaning it literally contained both nutrients, only the phosphorus was getting through.
Exactly. The plant was practically swimming in fertilizer, but it was actively starving for potassium.
Which is catastrophic for a plant's metabolic homeostasis. Potassium doesn't build structural components, right? It acts as a regulator.
Yes, its primary job is to control the opening and closing of the stomata.
The microscopic pores on the surface of the leaves.
Right. The stomata regulate transpiration. They open to let carbon dioxide in for photosynthesis, and water vapor escapes in the process.
And the potassium controls this.
Yeah, potassium ions pump into the guard cells around the pores, creating turgor pressure that swells the cells and opens the pore. Without potassium, the plant absolutely cannot regulate its water pressure.
So it basically loses its ability to manage hydration.
Yes, and it also loses the ability to efficiently transport carbohydrates from the leaves down to the roots.
So the Black Orchid at 36% phosphorus was literally suffocating. It couldn't balance its water, couldn't move its food, and just spiraled into deep physiological stress.
Exactly. The energy that should have gone toward blooming and producing flavonoids was desperately rerouted just to keep the cellular walls from collapsing altogether.
It's a stark reminder that in complex biological systems, more is rarely better.
Very true. Precision is the absolute dividing line between a thriving, medicinally potent organism and a completely stressed, failing one.
The margin of error is incredibly narrow. Finding that exact 14.4% sweet spot is what separates blind farming from true biochemical engineering.
It completely shifts how you view the survival of a species, doesn't it?
It really does. So what does this all mean for you listening? If we pull all this together, we started with the Black Orchid, right? An uncooperative diva on the brink of vanishing. And we looked at the mechanics of self-incompatibility, where the plant's own immune-like responses actively block its pollen tubes, basically forcing us to intervene.
And then we saw how scientists cataloged 30 structural traits and sequenced the rapidly mutating ITS regions of ribosomal DNA to identify C. rumphii as the perfect genetically compatible mate.
Right. By merging the sympodial architecture of these two plants, we bypassed the rejection alarms and secured a fast-blooming hybrid lineage.
And for the purebred native orchids still clinging to survival, we learned how to hack their ATP energy grid.
Exactly. By delivering precisely 14.4% phosphorus, we can force the stem cells to pump out gibberellins. This forces an early bloom and turbocharges the production of complex antioxidant defense compounds.
All while carefully avoiding the traffic jam of nutrient antagonism that would starve the plant of potassium.
It really demonstrates the incredible depth of science required to preserve the natural world. Maintaining a critically endangered ecosystem isn't just about, fencing off a rainforest.
No, it requires a granular understanding of ribosomal mutations and cellular transport proteins. Whether you're a casual gardener, a fan of botanical history, or someone interested in natural medicines, the effort that goes into this is just staggering.
Which leaves us with a really fascinating implication for the future. We've just explored how scientists can map the exact biochemical triggers of a rare temperamental orchid, perfectly tailoring a chemical spray to turn it into a highly concentrated medicinal factory on command.
And the applications of that kind of precision extend far beyond conservation.
Exactly. Think about the houseplants or the urban gardens we interact with every day. If we can dial in the exact percentage of macronutrients required to hack the metabolism of a wild orchid, what does this mean for the future of personalized pharmacology?
It's a wild thought.
I mean, imagine a near future where you aren't just watering a potted plant. Imagine scanning a common houseplant with a sensor, reading its exact hormonal and nutritional deficits, and applying a custom-tailored spray.
That would change everything.
We could essentially program common plants to overproduce specific defense compounds. You could be growing a highly precise, biologically optimized medicine cabinet right on your living room windowsill.
Suddenly, keeping a plant alive isn't a chore.
No, it's an exercise in biological coding. It's something to think about next time you water your ferns. Thanks for joining us on this deep dive.